Okay, just imagine this for a second. You get a phone call that is literally going to change your life forever. The kind of call every scientist on the planet dreams about. But it's 4:30 in the morning and the number is from some other country. What do you do? Well, if you're Dr. Mary Brunkow, the answer is probably not what you'd expect.
Yep. The call from the Nobel Committee, one of the most prestigious honors in the world, gets dismissed as spam. It's just a fantastic reminder that even Nobel laureates are human, and honestly, who doesn't value their sleep?
So this is the story of how that huge, monumental piece of news finally broke through. And it's not just a story about science. It's about the chaotic, kind of confusing, and just very real way these incredible moments can actually happen. So while Dr. Brunkow was still fast asleep, the world was already starting to find out. Her husband ends up finding the local news crew on their front porch, and apparently, the family dog had absolutely no idea why everyone was up before sunrise. You can just picture the scene, right? This is the moment where reality starts to crash the party.
And that's the real question, isn't it? I mean, beyond the funny story, there's this absolutely groundbreaking scientific discovery. A discovery so important that it fundamentally changed how we understand our own immune system. So let's dive into the science behind that call.
To really get this discovery, we have to go back, way back to the 1990s. Dr. Brunkow and her colleague, Dr. Fred Ramsdell, were working at a biotech company, and they were trying to solve this puzzle that all started with a very, very sick little mouse. So meet the scurfy mouse. Now this was not just any mouse. It carried this really devastating genetic mutation. Its own immune system, specifically its T cells, were just completely out of control, attacking its own body, leading to a really short, sick life. The team had to figure out why. What was going wrong? The challenge they were up against was just immense. I mean, today, you could probably map a mouse genome in a couple of days. But back then, it was, in her words, a molecular slog. We're talking about years of patient, painstaking work, sifting through 170 million DNA base pairs on just one chromosome to find one single tiny error. And they found it, the needle in the haystack. They discovered a gene that nobody knew about, and they named it FOXP3. Think of this gene like a master switch. When it's broken, like it was in the scurfy mice, or in humans with a similar rare disease called IPEX, the immune system just completely loses its brakes. It's all gas, no brakes. Now this discovery, it did so much more than just explain one rare disease. It basically kicked open the door to a whole new field of research by helping to answer a really fundamental question. How does our incredibly powerful immune system know not to attack, well, us? To really get the breakthrough, you just have to know the major players. So in our immune system, you've got helper T cells. They're like the scouts who identify threats. And then you've got killer T cells. They're the soldiers who go in and eliminate those threats. They are amazingly effective, but they're also incredibly dangerous if they aren't kept on a tight leash. This has been one of the biggest questions in immunology for decades. If your whole system is designed to seek and destroy, what gives the stand-down order? What prevents all that friendly fire against our own healthy tissues? And the answer, it turns out, lies with a special type of cell. The discovery of FOXP3 was the key that unlocked the role of these regulatory T cells. The Nobel Committee called them the immune system's security guards, which is a perfect way to think about it. And FOXP3 is the gene that basically trains and controls these guards. You take them away and it's just total chaos.
All right, so let's shift back to Dr. Brunkow's personal story for a minute. Because what makes all of this even more amazing is that a scientific career, even a Nobel-winning one, is almost never a straight line.
You can see it right here. The landmark paper gets published in 2001. But just a few years after that, the whole research site closes down, and her career path takes a turn. She becomes a science writer, then a program manager, working on totally different areas of research. A full 24 years go by between that discovery and the Nobel Prize call. Her humility is it's striking. In that early morning phone interview, she just straight up acknowledged that her own career has moved on from the very field she helped to create. It's such a powerful statement about how science and our own personal paths can evolve. And she makes one thing crystal clear. Science is not a solo sport. Throughout her interview, she just keeps emphasizing the collaboration. It wasn't one person's flash of genius, it was a collective effort. A whole team of smart people tackling a really hard problem together.
So why does all this matter right now? We've gone from a spam call to a sick mouse to a single gene. How does that fundamental research done decades ago actually impact medicine today?
While understanding FOXBOR3 and these regulatory T cells has opened up a whole new playbook for medicine. For autoimmune diseases, we can now try to boost these security guards to calm things down. For cancer, we can try to disable them, because it turns out tumors are clever. They use these T cells as their own bodyguards. And for organ transplants, they can help prevent the body from rejecting a new organ.
Ultimately, this whole story just shows you why that basic, fundamental science is so critical. The patient, difficult work of figuring out what was wrong with one little strain of mice ended up creating an entire new field of research, and now it's leading to treatments that could save countless lives.
And that really leaves us with a final thought. Dr. Brunkow's Nobel came 24 years after her paper was published. It really makes you wonder, what other world-changing discoveries are just sitting there right now in an old lab notebook, waiting for the rest of the world to catch up?